Hello and welcome to this video. In this video I want to talk about something called the Jupiter Icy Moons Explorer project, or JUICE for short. Very interesting project, and its main objective is not actually to find life but to see whether there are conditions which would support microbial life on some of the icy moons of Jupiter. It's a fascinating subject, and it's kind of linked with the idea that life should be existing on other planets besides planet Earth, and if there are the right conditions, we would expect to find life elsewhere in the universe. Although the JUICE mission itself is very clear that it's not looking for life, it's looking for conditions which could support life. And this is very interesting, but I think it's a bit of a lost cause, because it's all very well to say we might have conditions which support life, but we don't really know how life arose on planet Earth. We don't have an explanation of how life arose spontaneously on the Earth. And this is because life is in fact very complicated. So I'd like to just go through some of the issues around the origin of life and whether it could actually arise spontaneously without the intervention of an intelligent designer of some sort. So life requires certain things. There has to be catalytic molecules. There has to be information storage molecules. Proteins are the catalytic molecules that we know of, and DNA and RNA are the molecules that store information. But the question is, can these things arise spontaneously? And if we have the right conditions anywhere in the universe, would we expect life to arise spontaneously without the intervention of an intelligent designer? Another molecule that is important in the whole question of life and how it could arise are molecules called ribonucleoproteins, which are a combination of a protein and RNA, and they could in theory do both jobs, the catalytic job and the information storage job. To understand a little bit of the problem of life arising spontaneously, by which I don't mean it has to arrive very quickly, the current scenarios involve some sort of stepwise series of spontaneous events which result in chemistry becoming biology. Back in the 50s and 60s, there were a series of experiments done to try to demonstrate how this might be possible using supposed conditions on an early Earth, the Miller-Urey experiments and others following on from that. And they were looking to see whether we could get the chemicals of life through a process which could occur without any intervention. So they didn't manage to produce DNA or RNA or protein. They didn't even find a route to get to the lipids that are involved in life as well, and the carbohydrates. All of these things which are needed for a cell, they were not able to demonstrate that. All they got was some very simple amino acids and other simple molecules. There are various scenarios which are proposed to explain how life could have arisen spontaneously, one of which is protein first, because of the difficulties with getting DNA and RNA first. The problem with this is that amino acids that make peptides only do so in the absence of water. Spontaneous sequences that are generated when you mix amino acids together in water are different from functional proteins, because not all the possible bonds between amino acids are peptide bonds. It's only a certain type of bond which can produce a functional peptide or a functional protein. And in fact, some people have suggested that in order to get one working protein by chance, in other words, without intervention by an outside organizing agent, every molecule in the universe would have to have been an amino acid. So people have suggested that an alternative scenario is RNA first, ribonucleic acid first. Ribonucleic acid and DNA are similar molecules. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. The advantage of starting with RNA is that RNA can actually be an enzyme or an information storage molecule. So this scenario is very popular. Is an RNA first model likely to work? Well, the first problem is the formation of the ribonucleotides needed to make RNA, which requires ribose, phosphate, and 4 different bases. Currently, there is no known mechanism for 2 of these bases. Ribose also is not known to form spontaneously. Another problem is, even if you could overcome the problems with getting together these various molecules, RNA is less stable than DNA and is not going to hang around very long waiting for the protocell. And in fact, all the RNA we know of is made by protein. So given these problems with the RNA world, people have proposed alternative models involving something called RNA ligase molecules, which are, again, functional in the catalytic sense and can also be autocatalytic. The problem is most of the functional ligases are over 100 nucleotides long, and they are composed of approximately equal amounts of the four bases, adenine, uracil, guanine, and cytosine. And there is no known mechanism to ensure that you get the equal amounts of these bases in the right place at the right time to form a functional ligase. And another problem is that ligases are very unstable and break up in water, and you need mechanisms to preserve them. So basically, the problem is that we live in a world where life is made up of DNA, RNA, and protein, and there are various difficulties with getting to this point because proteins are made from instructions coded in DNA. Proteins are needed to read the instructions to make the RNA that is read by other RNA to make proteins. And DNA is repaired and copied by proteins. So there is a big problem getting all these things together in the right place at the right time. And part of the problem is the conditions required to make the various precursors for proteins and nucleic acid. The conditions required for amino acid formation are different from those required for activated nucleotides. So you're going to have to come up with a scenario where amino acids can form in one place and activated nucleotides in another place, and then bring them together at the right time to actually produce life. Given all these problems, people have come up with various other scenarios, one of which is the idea of self-organization. One of these ideas is that geothermal metal sulfides could provide a route to life. Another is crystals on clay can help organize the various molecules into biomolecules. And then there are these things called hypercycles, which are being proposed as a solution to this problem. The trouble with all these is that they are all hypothetical. Just consider hypercycles for a moment. A hypercycle is a theoretical prebiotic cycle of chemical reactions involving the autocatalytic amplification of substances thought to have been required for the presumed spontaneous origin of the first life forms. Well, all this is very hypothetical. And the big problem is actually there are no examples of working hypercycles. So to conclude, there is no known spontaneous mechanism by which life could evolve, even given conditions under which life can survive. So this is why I would suggest to you that the JUICE mission is very interesting and exciting, but unlikely to come up with any substantial solution to the problem of life and how life is sustained. We come back to the original point that I was trying to make, that life requires an intelligent designer. And I would like to suggest to you that the intelligent designer is the God of the Bible. Thank you for listening.